Hello. I can figure these things out.
Are we doing clicker today? Because mine just said.
We're not doing clickers and we're not doing them for the quarter.
Okay.
Thank you very much. You get to see the double team. I brought my Aussie buddy Mike with me. So.
That's fun.
Do you think there are many more students coming? Or
There are a few more coming, but this is pretty close.
Let's roll.
G'day guys and greetings from Australia, my name's Mike, this is James from International Student Volunteers Down Under. Just want to tell you guys, if you ever want to go travelling overseas to places like Australia, Europe, Africa, South America, or Thailand.
We're going to be selecting about 50 students from UC Davis to represent America on a special student work program this summer.
Now, if you're selected to participate, you'll have the opportunity to receive academic credit while you're travelling overseas with a group of students for 1 month during the summer.
Guys, the first 2 weeks is a volunteer project. We have one of two choices. You can choose to work in social community development, working with underprivileged children and families in places like
the Dominican Republic, Thailand, East Europe, and Costa Rica. We do things like teach English to children, build schools and houses, and on some projects you guys will actually get to live with a local family.
Or guys, you can work in conservation, working in environmental areas like rainforest regeneration and endangered species management with elephants, dolphins, and cheetahs in places like Australia, New Zealand, South Africa, and Ecuador.
During the second 2 weeks, we'll travel all around your host country, meeting with the local people whilst exploring various ecosystems. You'll also participate in some awesome adventure activities like whitewater rafting in Costa Rica, glacier climbing in New Zealand, scuba diving in the Great Barrier Reef, and wildlife safaris in Africa.
Guys, ISV is awesome, and we were recently selected as one of the top 10 volunteer programs in the world. The best part by far is that most students pay for the cost of the program through tax-deductible sponsorship donations from the community.
So guys, if this sounds exciting to you, we're going to be passing around these bits of paper. Make sure all of you guys put your name and email clearly so we can send an email later today with more info. Then guys, rip a flyer off the back. That'll tell you all about the information meetings we're having all day tomorrow in the NU building, 3rd floor, meeting room, every hour on the hour from 9 to 5. That's
9, 10, 11, 12, 1, 2, 3, 4, and 5 o'clock where you can meet other students from UC Davis who traveled with us last year.
You guys have an absolutely awesome opportunity to come travel with a purpose, meet some amazing people, do some awesome adventure activities, and make a real difference. We look forward to seeing all of you guys at the meeting on Thursday. Thank you very much, Professor, and we'll see you at the meeting on Thursday.
Thanks again, I appreciate it.
And we'll collect those bits of paper up front at the end of class, guys.
On this table right here. Thanks again, Prof.
Yeah, sure. No. Start the excitement out. Sounds cool. Did you get it at the top? No. Yeah. All right. Brian Moore and I are going to now do the same thing for phylogeny up front. So. I sometimes get complaints that I'm too loud, so I'm trying to encourage people like them to come in first. So I seem like I'm subtle or reserved and shy. All right, so what we're going to do today is take the next step in looking at evolutionary reconstructions, and we're going to go into how you actually figure out the tree itself. Prior to this, we've talked about what a tree looks like, the different parts of a tree, and how we trace the evolution of characters on specific trees, so assuming that we're given a tree. But what we're going to do now is talk about what we do when we're not given a tree. So again, in the previous lecture, we focused on characters and character state, reconstruction. Today, we're going to talk about what's called phylogenetic inference. Inference basically means you're trying to figure out what the phylogenetic patterns are. And we're going to talk about this in at least part of the next lecture, too. So there'll be a little bit of run over in phylogenetic inference into the next lecture. What I'm going to do today is take you through basically these four topics. Just a brief introduction to sort of the concept of phylogenetic inference. A little bit of a detour, although you'll see why it's relevant, to talk about a particular type of phylogenetic tree called an unrooted tree, which is going to be very important for what we're doing with phylogenetic inference. Then we're going to talk in specific detail about how you actually do phylogenetic inference. And the last part we'll probably get to in the next lecture, which is taking an unrooted tree and then actually rooting it once you're done with the phylogenetic inference. So, as I've said already, and I haven't been completely explicit about this, but we've assumed in every one of the cases previously, basically, that we were given a tree. So I showed a tree of A, B, C, D, E, F, G, H, etc., and we then analyzed some feature, either monophyletic groups or character evolution on that tree. But most of the time, we're not given a tree, we're given taxa. And we can measure some data about those taxa, and then we would like to figure out what the correct tree is, showing the relationships among those taxa. So here's an example of what it might look like from a phylogenetic tree perspective. If you don't know the evolutionary history of organisms, this is you could call this a massive polytomy. This is, with these taxa, and we know nothing about the relationships among those organisms. You can also use phylogenetic inference to figure out relationships among organisms if you know some of the patterns in the tree, but not all of them. We're going to focus on cases for now where we don't know any of the patterns in the tree. To make it a little less boring, although I'm sure it's still a little bit boring here, we're going to use actual pictures of organisms some of the time instead of letters, but it's the same thing. Can be applied to any taxa across the tree of life, this method that we're going to talk about. So rather than make it really complicated, if we were trying to do this with 8 or 9 or 10 taxa, it would take us many, many hours to do this exercise. So we're going to focus on just 4 taxa, those 4 that I've highlighted here. We're going to throw away the other taxa in the tree. Now again, remember I talked about when we show a phylogenetic tree, it's an oversimplification of evolution. When we take out these other taxa from our analysis, it doesn't mean that they don't exist. It doesn't mean that they don't have interesting phylogenetic patterns related to these organisms. What it means is we're only trying to figure out the relationships among these organisms. The other ones are still there somewhere in the big tree of life relative to these taxa that we're looking at. We're just not going to include them in this analysis. Now we're going to do one other thing, which is going to be a little bit sort of the most conceptually difficult thing that I've done in the class and that I might do in the entire set of lectures that I'm doing, which is we're going to pluck off the root from this tree. So all the trees that I've shown so far have been where there's a root going into the root node, and that root node represents the common ancestor of all the organisms in the tree. We are going to now take off that root and have what's called an unrooted tree. And I'm going to take you through some different ways to think about unrooted trees so that you will understand, at least in part, what they mean. But this is a sort of big conceptual leap that we're doing here. Unrooted trees are a theoretical construct. So evolutionary history of organisms, we think that any organisms that we're comparing have a common ancestor somewhere, and that there was a root branch leading into the common ancestor of those organisms. But for many purposes, we don't want to include that root in our phylogenetic analysis. Just like with any of the other branches, any other trees, we can swing around the branches in unrooted trees, as you will see in a minute. For this unrooted polytomy, I can do whatever I want with those taxa, and it all means the same thing. There's a polytomy with many taxa coming out of this single point here. So there are a lot of reasons why you would do something. You would want to do something in what we call this unrooted tree form, and I've outlined some of them here. These are examples of the reasons. There are many others. So when we root a tree, I've shown you this already, there's frequently we show an in-group. Those are the groups of organisms that we are interested in studying the evolution of. That could be these 4 taxa. And then an out-group. And the out-group is a lineage or multiple lineages that branched off in the evolutionary history prior to the existence of the common ancestor of the organisms in the in-group. And that helps us determine where the root of a tree is, as you will see later in the class. But many times we don't have a good candidate for an out-group for a group of organisms. You may not have any out-group for the organisms that you're looking at. So you may not be able to use the out-group approach to root a tree, so you have to work with what are called these unrooted trees, as you will see in a minute. Another reason why you might want to work with unrooted trees is you may have a candidate out-group, but it may be essentially useless for the type of information that you want to analyze. So, for example, in the lab, you do phylogenetic analysis using this out-group method that I'll talk about again in a minute or two, where you compare the character states of organisms, and you use the character state of the out-group to determine what the ancestral state is for your organisms in the in-group. Well, what if you had 3 organisms in your in-group, like primates, and for whatever reason, the only other organism you could compare to for the out-group was slugs, and you're interested in brain evolution? Well, the features that you're comparing may not be very comparable when you're looking at an out-group of slugs. They may not have any of the character states that you're looking at. They may be useless for looking at ancestral character states in your group of organisms because they might not have any of those features. So that reason is another reason why you might not want to not be able to include an out-group in your analysis. What you're going to see today is another reason, this third reason here, which is that for many analyses that we would like to do, unrooted trees are actually simpler to use and to analyze than rooted trees. And I know you might not understand this right now, but I'm going to show you why that is the case. So what I'm going to do first is show you what unrooted trees look like in a few different ways to get you to start thinking about them. This is the way we would draw one example of an unrooted tree for 4 organisms. I'm not showing polytomies here. So for these 4 organisms, I've drawn this unrooted tree, and just take it as a given for now that this is what an unrooted tree could look like for those 4 organisms. The challenge here for thinking about it is that the root in this tree could be in many different places. And depending on where we place this root, it will influence what you think about the evolution of these organisms. And I'm going to show you the ways to root this tree in a minute. But I'm going to show you it backwards first, so you just start to think about what an unrooted tree means. So on the left is a rooted tree of these same 4 taxa. And I'm going to pluck off the root from this tree and show you how to convert it into this unrooted tree on the right. So I'm taking off the root from the tree, taking off the root node from the tree. This now, even though there's a little kink here, there is no node there left. We've thrown away the root node. So we have to straighten out that branch in essence so we don't see any splits in that branch. And rotate around these taxa there, and we get the same tree that I showed on the right. So we've removed one of the branches and one of the nodes, and then just played with the rotation to show you that you can convert a rooted tree into an unrooted tree. So now what I'm going to do is we're going to talk about, starting with an unrooted tree, how can we convert it to a rooted form by placing the root in different places on the unrooted tree. So again, here's one form of an unrooted tree for these 4 taxa. And it may seem to you, if you look at this, that A and B are what you would like to call sister taxa. That is not true in some of the cases here. And I'm going to show you, depending on where we place the root on this tree, A and B may not be sister taxa. And the same is true for C and D. So we can place the root of the tree into any of these branches here. So I'm going to place the root of the tree into the middle branch here. And then I'm just going to, in essence, convert this tree now into a vertically drawn tree by rotating some of the branches. So I've rotated this big set over here. There's a branch going to a node, and then coming off one node is A, coming off the node in one direction is A, and in the other direction is B. That's the left half up here. And I've done the same thing on the right half. There's a branch going up to a node, and then C and D coming off of that node. So for this particular rooting of that unrooted tree, A and B are in fact sister taxa. And C and D are in fact sister taxa. Do you see what I did with rooting this unrooted tree there? Okay, so now let's root it in a different place. If we root it here, say the root of the tree is on this branch, we're going to do that same unwinding activity. So I have the root, the red branch here, and then coming off of one side of the root is going to be B. And then coming off the other side of the root is a branch that's going to go up to a node. And from that node here, one branch is going to go up to A, and another branch is going to go up to the common ancestor of C and D. We're literally just walking our way through the tree and redrawing it as a vertical tree. And you can see on this tree, when we draw the root on the branch that is between B and this node, now A and B are not sister taxa. The root of the tree has split up A and B over here. We can do the same thing, drawing the root onto the branch connecting A to this node, and it's basically going to look very similar to this tree, except with A coming off of the bottom. So here, A, and then something going up to a node, B coming off one side, C and D coming off of the other. So you have to start thinking about this. When we draw an unrooted tree, the key thing is you can't figure out what are sister taxa because there is a branch and a node missing from that tree. And the only way things can be sister taxa is, in essence, if they are connected by a single node to their common ancestor. So you can't immediately figure that out, whether or not they're sister taxa, until you figure out where the root of the tree is. Does that generally make sense to people? Any questions about this? Okay, so this is an example of a type of question we might ask you on a test, although some of these might be harder, some of these might be simpler than on the test. So for this tree, how many possible rootings are there for this tree? How many possible places can we place a root in this tree, assuming there are no polytomies? Right, so 5. There are 5 branches in this tree, and we can put the root in the middle of any of those branches. Each one of those roots creates a different rooted tree. Every place that we place the root will create a unique rooted vertical tree for this group of 4 organisms. So I'm going to ask a question which is much harder. We would not expect you probably to make this immediate transition on a test, but I wanted to force you to think about this before talking about what the answer is. So if you have 4 taxa, how many different possible rooted trees are there for 4 taxa? All the possible ones, not just the ones that I just drew. So with 4 taxa, how many different possible rooted trees are there?
24.
Why do you say 24? It's close, but that's not actually, it's not directly factorial to calculate how many trees there are. So maybe you should play with this on a piece of paper. You should certainly go home and try and figure this out. I'm going to show them to you now anyway, but you should play around with seeing if you can draw all these different trees. So here's one vertical tree showing A and B on one side of a major split and C and D on the other side. We can draw a few other versions of this tree with a split down the middle in essence. So we can draw A and C on one side and B and D on the other, or A and D on one side and B and C on the other. There are no other ways to draw 4 taxa with 2 on one side and 2 on the other, because we can just rotate the branches to create every other pattern you could think of in your head. You can have A and B together, A and C together, or A and D together. So those are the 3 possible trees where there are 2 sets of 2 together. So now let's draw the trees where A is on its own as the first branch off. If A is the first branch off, you can have B on its own or C and D together. You can have C on its own and B and D together. And you can have D on its own and B and C together. Again, you can rotate these branches to create sort of other patterns, but the key thing to do is focus on the groupings here. We can do the same with B being on its own, branching off first, and then A can be the next sort of the unique branch that's left with C and D together, C on its own with A and D together, or D on its own with A and C together. The same thing with C being that deep branch, and the same thing with D being that deep branch. So there are 15 unique trees for 4 taxa, unique rooted trees. And the main reason I'm showing you this is now we're going to compare that pattern with how many unique unrooted trees are there for 4 taxa. So how many different ways can you arrange the taxa on this particular tree structure? Because this, in fact, is the only tree structure we can have for an unrooted tree with 4 taxa. How many different ways can we arrange those taxa there that are unique from each other? Right, so who said 3? Can you explain, say what those patterns are?
Well, A, B, C, D, or A and C, B and D, or A and D, and C and D.
Exactly. So there are only 3 possible ways we can arrange 4 taxa on an unrooted tree. We can have A and B together. That immediately forces us to have C and D together on the other side. We can have A and C together. That forces us to have B and D together on the other side. Or we can have A and D together, and that forces us to have B and C together on the other side. That's it. There are only 3 possible unrooted trees for 4 taxa. So many of the methods that people would like to use to infer evolutionary history require us to sort of take a tree and map some type of features onto the tree, like character states. It's going to be a lot easier to do that with 3 trees and to compare them to each other than with 15 trees. So we're going to take you through how to figure out phylogenetic relationships among organisms using unrooted trees, in part because they're just easier to deal with the same number of taxa. We have fewer things to analyze. You should try drawing these different forms. You should try from scratch drawing the 15 different rooted trees for 4 taxa. And if you can do that, most of what we're going to do for phylogenetic inference in the class will make a lot more sense. Okay, again, here are those 15 trees versus the 3 trees. Much easier to deal with these 3 than those 15. And what we're going to do is split up the tasks. We're going to analyze unrooted trees, and I'll explain to you how we're going to analyze them in a minute. And then after we analyze unrooted trees, we're going to place a root on them. Yeah?
How many possible for the 4 for the rooted ones?
Yeah.
Why is it 15 instead of 16?
What is the 16th?
I feel like the 16th would be, I feel like it would be all of them like on their own and none of them be like sisters. Never mind.
Try and draw it. I don't think there is another one. All theory suggests that there should be 15. So probably what you're thinking in your head is a version, a rotated version of one of these trees that's already drawn. But we can talk about it later, or we can try drawing it on the board later or something. I'm I'm certain that there are only 15 different versions. If we have polytomies, you can have other versions. But without polytomies,
So one way to think about it is if there's n species and n is 4.
Yeah.
So the difference between 3 and 15 is a factor of 5, right? And so the reason for that is on the unrooted tree there are only 5 branches.
yeah.
And there are 5 places to root it. So in general, there are 2n minus 3 times more rooted trees than there are unrooted trees for n species.
It's the there is a mathematical formula for this. It's not factorial. It's not obvious to go directly from the numbers. It's a slightly more complicated formula. But he is right. Another way to think about this is there are 3 unrooted trees. And for each of those unrooted trees, there are 5 places we can root those 3 trees. And each of those 5 rootings creates a unique rooted tree. So 3 times 5 is 15.
And there's mathematical proof.
Yeah. That if you can prove otherwise, you will win a major evolution prize. So I'd love to see it, but we don't think it's true. Yeah.
So you already said that the last slide, the one with the three, yeah, that one, not that one, the last one. Yeah. You said that we can't establish sister taxa based on an unrooted tree.
Yeah.
So how do we know there's only 3 depending on the groups of A and B and C and D? A and C and B. If they're not sister taxa.
They're not sister taxa. There are only 3 ways to put any taxa on the edge of a drawing that looks like this. That are not. Now remember, we're not including the root here. There are only 3 ways to do this, but if we rooted them, they could be different. So that's why there are 15 different rooted trees, but only 3 unrooted trees. And this makes it easier to do some analyses because in many cases, all we have is information about modern taxa. And those we can place at the end of the tips of this unrooted tree. We very rarely have real knowledge of the root. And so for doing analyses like this, it's easier to just analyze the tips as independent things without adding an extra branch into the tree that we're proposing existed, but we know nothing about.
The shape doesn't imply sister taxes.
It doesn't because the root could come in here and then A and B are no longer sister taxa. And the root could come in here and then D and C are no longer sister taxa. But this particular tree, A and D could never be sister taxa, no matter how we root this tree. They are already separated by 2 nodes. And that means for this particular pattern, no matter how we root it, it is impossible for A and D, or A and C, or B and C, or B and D to be sister taxa. And you should root all of each version of these 3 unrooted trees you should root, and you will see that being proven to you. There was another question somewhere? Okay. Oh, sorry. Right, so I said when I asked the question that we're not going to allow for polytomies. If the root came into there, it would now have 3 branches coming out of that root, and that's a polytomy. So in theory, actually, that could happen. It would make it even more complicated. I'm making this simpler so that it doesn't make our work a little bit harder in a minute as we try and figure out what the possible trees are. So polytomies are possible. We usually don't look for them unless there's some evidence for them. But you're absolutely right, that could be done. Okay, so here are those 3 trees again, the 3 possible trees for 4 taxa, 3 possible unrooted trees. We're going to replace those with the organisms that I showed you, the 4 organisms that we were going to study: wasp, mantid, beetle, and fly. So here's one tree that has wasp and mantid on one side and beetle and fly on another. Here's one that has beetle and mantid together, wasp and fly on the other, and here's one that has fly and mantid together. These are the 3 different unrooted trees. Yeah?
When you have the wasp and the mantid there, that doesn't mean they are sisters, right?
It doesn't yet, because the root could come in between and split them up.
So in this kind of trees, we don't see their sisters.
You can't. You cannot say with an unrooted tree what are sister taxa. You can say what cannot be sister taxa. Because in this tree at the top, wasp and fly cannot be sister taxa. There is no way to place a root on this tree such that wasp and fly would be sister taxa. But since we don't know where the root is yet, we don't know if wasp and mantid are sister taxa.
The only way is by putting the root, then we'll see.
Yeah, and we're going to see that later. Okay, so now what I'm going to do is, in the next 20 minutes, is walk you through an exercise of figuring out, given a set of data, which of these 3 trees is the best tree, is the tree we want to use for studying the relationships among these organisms. So this is a pretty common task in evolutionary biology. It's based upon a very straightforward principle. That is that organisms accumulate differences from each other once their lineages split. The more recently organisms shared a common ancestor, the fewer differences they should have. Now, that's not always the case. We know that there's convergent evolution in some features. But on average, the more recently they share a common ancestor, the fewer differences they should have because there's been less time for differences to accumulate. And you can then compare the patterns of similarities and differences among organisms and rank them in some way. And you should be able to figure out what the relationships are among those organisms by analyzing their patterns of similarities and differences. Now, it turns out there are hundreds of different approaches to doing this in the evolutionary literature. We can classify those approaches into major categories of approaches, and there are, in fact, probably 5 to 10 major categories of those methods. I'm just going to quickly explain 3 of those categories to you before we go into one of them in more detail. They're all based upon that same principle that I just told you, comparing similarities and differences among organisms and trying to use the patterns of those similarities and differences to figure out the order of branching among organisms. But they differ in how they do that. So the parsimony methods, which are an extension of the character state reconstruction method that we already talked about, that used parsimony. Parsimony methods try and find the best tree by looking for the tree that has the fewest number of changes in character state over evolutionary time. And we're going to focus in on this method in a minute, so you'll be sick of this approach very shortly, probably. That's one category of methods. A second category of methods are called distance methods. They do a little trick that works in some cases well, doesn't work in other cases well. They take the features that you're comparing among organisms, and they convert the overall similarity of those features into a distance, a dissimilarity between organisms. And you create a matrix of this difference between organisms. So you compare organism 1, 2, 3, and 4 to each other, and you see how different they are from each other. And then these methods try and come up with an evolutionary tree where the lengths of the branches most closely reflect those distances that you calculate. And then there's a third class of methods. I mean, again, there are many classes of methods. But a third major class of methods are called likelihood methods. They are similar to the parsimony methods in that they try and give a score to different trees. But unlike parsimony, the likelihood methods, the winning tree is one that has the best statistical probability of producing the data that you observe. So you actually create a statistical model of how traits change over time. And then you apply that model to each tree. And you ask, given this model, what is the probability that I could generate the actual data I observe, given tree 1, tree 2, and tree 3, and tree 4, and so on, if you had more. So it's a statistical approach and has a lot of power from being a statistical approach, as opposed to parsimony, which is a counting approach. We're going to talk about each of these methods a little bit throughout the rest of the class, but for the rest of today and much of Friday, we're going to talk about just the parsimony method to teach you about the principle and how you go about doing a form of phylogenetic reconstruction. So we've already talked about, yeah, question?
Yeah, I was wondering, so the likelihood method is similar to, I'm imagining like if we were to scan the DNA of two organisms and try and see how similar they are and then compare all that data and go through, is that sort of what?
Yeah, so it turns out that the likelihood method, so the question was asking if likelihood methods are, sort of applied to DNA data and comparing DNA data. It turns out that for likelihood methods to work well, you need to have a good model of how traits change between organisms. And we have a pretty good idea for how DNA changes between organisms and even protein sequences change between organisms. Likelihood methods work really well in cases where you can build such a model. They're a little bit trickier to apply in cases where you have things like bone structure. And you might not know how to create a model of how the humerus bone changes between taxa. But you could apply any of these methods to any type of data. It's just some work better for certain types of data. Likelihood methods in general, most modern phylogeneticists tend to prefer because they bring the power of statistics. However, they can be computationally challenging. They require sometimes a lot more calculations than other methods. So I'm going to focus on teaching the parsimony methods because it'll teach you the principles, and we can go through it a little bit more easily than we can go through the distance or the likelihood methods. But I will talk briefly in the next lecture about one of the reasons you might want to use likelihood methods over parsimony methods. Okay, so we already talked about this, taking a given tree and then trying to figure out what the history of characters was on that tree for things like flight, recreating the history of ancestral and derived character states by finding the reconstruction that required the fewest number of changes. That was given a specific tree. We are going to do the same thing here, except we're now going to apply this across many different trees instead of just looking at one tree. So it's the same principle. We try and find the tree now that requires the fewest changes, as opposed to just one character reconstruction on one tree that requires the fewest changes. And I'm going to walk you through in detail how to do this in the next 15 minutes. So I just want to take one step back before we do this, and I've already mentioned this today, but I want to do it again. So in lab, you use a method that's generally called the outgroup method to look at character evolution and even to figure out what the best tree is. The outgroup method uses a trick which says that if you have multiple character states in your in-group and you have an outgroup, you can assume that the ancestral character state is the state that is in the outgroup. It's a trick. It is a trick that works in some cases and doesn't work well in other cases. I prefer to use the unrooted approach because it doesn't require making any specific assumptions about what was the ancestral character state. But they're both used and they both have their uses. So we do one in lecture and one in lab. So we're going to focus on this unrooted approach in lecture. And again, it is distinct from what you've been doing in the lab for most of the parts of the lab. So here's a summary of what we're going to do. This is, in essence, it. But we're going to have to go through all the details because the devil is in the details here. We're going to take the set of taxa that we want to study the relationships among, and we're going to draw all possible trees that show the relationships among those taxa, all possible unrooted trees. And then we're going to compare a bunch of different characters between those taxa. And for each character, we're going to count the minimum number of required evolutionary changes for each tree. So we're going to, character 1, we're going to compare, say, for tree 1, how many character state changes are required. For tree 2, how many character state changes are required. For tree 3, etc. We're going to do that then for character 2, character 3, character 4. And when we're done, we're going to sum up the minimum number of changes required for all the characters that we're going to look at. And the winner, the tree that we're going to pick as being the most parsimonious, is going to be the tree that requires the fewest total number of changes across all the characters. So what are the characters we're going to look at? It could be anything. Again, these methods can work with any type of data. So we could use presence and absence of wings, different types of eye color, leg length, number of antennal segments, or DNA sequence data. We're going to convert that data into some sort of code like you did in the lab because it makes it easier to analyze than having to write leg length and wings, etc. over and over. Usually for morphology, we convert it to this binary 0 and 1 or 0, 1, 2 code. For DNA data, we just use the abbreviation of the DNA base. And then we create a matrix where the rows are the taxa and the columns are the different characters. And in each box, we write the character state for that character for that organism. So here's an example of a data matrix with morphological characters and DNA characters for those 4 organisms that we want to study the evolution of. That's a little too complicated for going through it in class, so I've created a matrix that's just DNA data, rather than switching back and forth between different types of data. We're going to do it just for DNA data. So we're going to use this data right here to figure out which of the trees showing the relationships among these organisms is the best. So here are those 3 unrooted trees for these 4 organisms. And we're going to go to character 1 and ask, for character 1 on tree 1, how many character state changes are needed for that character on tree 1? So here's character 1. We're going to convert the taxa tree to a character tree. Doesn't matter what the organisms are here. We're just trying to figure out, for this pattern here, that's tree number 1, what is the minimum number of character state changes required to get the characters we observe for that tree. So let's zoom in on tree 1 for character 1, and here it is. And what we have to do is test all possible character state reconstruction patterns. We have to give the nodes that are not labeled in this tree labels and ask how many state changes are required in this tree for that particular pattern. So for this pattern, for these 2 nodes here, we can create 4 plausible patterns for those 2 nodes. Either C and C, C and T, T and C, or T and T. We don't add in the other 2 DNA bases because if we added them in, we'd require extra changes. So we know that the nodes here have to be in one of the states present in the tips. That's just like what we did with bird flight and bat flight. So here it is. I've taken that tree and drawn for the nodes each of the possible patterns for the character state for those nodes. Here's one possible pattern, C and C, C and T, T and C, and T and T. Those are the 4 possibilities. And now we're going to ask which one of them requires the fewest number of changes. So this one requires 1 change. You look for any difference between 2 points in the tree. So that requires 1 step. This one requires 2 steps. This one requires 4 steps. And this one requires 3 steps. So these are 4 possible character state reconstruction patterns for character 1 on tree 1. The one that requires the fewest steps is this one. If we chose this pattern, we would only require 1 change in character on this tree. So that's what we pick. We want to find the minimum. So now we say that the minimum for this character on this tree is 1. And now we go and do it again. We do character 1 now on tree 2. So we take this tree. We do the same thing that we did a minute ago. We create the 4 possible patterns for the nodes. We ask, what are the minimum number of changes required for these patterns? Here it's 1. Here it's 2. Here it's 3 or 4. And here it's 3. So again, the winner is 1. So for character 1 on tree 2, the minimum that we need is 1 character state change. Now we do it again. Character 1 on tree 3. You can probably guess what the minimum is going to be. You can create the model in your head. Anybody guess what the minimum is going to be? 1, right. So it has the same pattern generally. There's 1 organism that has something unique, and everything else is the same. You can create a tree that a character reconstruction that has just 1 change for that. So you've already sort of figured it out for character 1 on tree 3. Now we have to go, so we sum it up here, give it a score. Now we have to go to character 2. Here's character 2. Anybody guess what the number of changes required for character 2 on tree 1 is? 0. Tree 2? Tree 3? Very good. So there are no state changes, no differences among the tips. So the minimum is going to be 0 changes. Now we could have created a model where there were changes, because we could have had the nodes being A. But that's not going to be the minimum. The minimum would be that both nodes are T in that tree, so we don't require any changes. Right now, all the trees are tied. The sum for each tree is 1. Let's go to character 3. This is the Gaga tree. So character 3, that was not on purpose, by the way, but I'm happy that it worked out this way. So character 3 now we're going to map, again, onto tree 1. The 4 possible patterns are AA, AG, GA, or GG. Now let's count. For that tree, there are 2 steps required. Sorry, for that pattern. For this pattern, there are 3 steps required. 3 for that pattern, and 2 for that pattern. We have a tie. We don't actually care which one of these wins. All we care about is that the minimum is 2. So we're going to give that character 3 for tree 1 a score of 2. Could be 2 changes there or 2 changes there. They're equivalent in terms of the score. Now we're going to do character 3 for tree 2. Those are the 4 possible patterns. There's 2 steps, 5 steps, 1 step, and 2 steps. So for this character on this tree, you only require 1 change. That's the minimum possible. So we give that a score of 1. We go and do this for tree 3 now, for character 3. It's going to be the same pattern generally as tree 1. So 2 of them will require 3 steps, and 2 of them will require 2 steps. I should have highlighted the 2 steps there, obviously. 2 steps wins. We give it a score of 2. It could be either of these patterns, but it gets a score of 2. So now we have a difference between the trees. One of the trees requires 2 steps. 2 of them require 2 steps. 1 of them requires 1 step. So now we continue. We do character 4. Character 4 requires 0 changes. You should practice these at home because I'm not going to walk you through the rest of them. Character 5, each one requires 1 change. Character 6 has the same general pattern as character 3. Mantid and beetle have the same state. Wasp and fly have the same state, and it gives you the same score. Tree 2 gets 1. Tree 1 and 3 gets 2. Character 7, tree 1 is actually better for character 7. And you should work that out playing with that at home. Character 8 is 0. Character 9, everything gets 1. Now we sum up across all these characters. Tree 2 requires 7 steps. Tree 1 requires 8 steps. And tree 3 requires 9 steps. So we have a difference between the trees. Using the parsimony approach, we are going to pick tree 2 as the winner here. So does that basically make sense, what we've done here? For each character, we have to give it a count for each tree. What are the minimum number of changes? And we walk through all the characters. Now, on a test, we're not going to expect you to solve this for, say, 200 characters in this way, but we do expect you to be able to do all of these recreations. And what I'm going to just introduce you to and then talk about more on Friday is a little bit of a trick for figuring this out, which you probably already may have started to notice in this data. There is something called parsimony informativeness. This is whether or not a particular set of a character and the character states for that character are going to be informative for distinguishing among different trees. So you saw some of the characters were identical and everything. There is no information there that we can use to distinguish among the different trees for characters that are identical to each other. So this parsimony uninformative are the characters that don't tell you which tree is a better tree. So an example is character 2, where everything was identical. Again, the minimum number of changes was 0. And we can go through all the characters and find these and block them out. Sorry, I'm just going to skip ahead and we'll come back to this on Friday. So there are 3 characters that are identical to each other across all the taxa. We can just ignore them. We don't have to include them in our analysis. Yeah? What about the ones that are all 1? That's a good question. That's the next thing we're going to talk about. The ones that are all 1 also are not informative for parsimony reconstruction because all trees will get a score of 1 because the change could occur at the tip of the branch on any tree. So we're going to throw those away too. And in our analysis, the only ones that are left are these 3 characters. If you had 200 characters, you could quickly scan through them and figure out which ones are going to be informative and which ones are not using this approach. What I'm going to explain in the next lecture is this rule that I'm showing up here, and we'll come to it in the next lecture, that to figure out if something is parsimony informative, there have to be at least 2 character states. That is, there has to be some variation. And each of them has to be present in at least 2 of the taxa you're studying. And we will come back to that on Friday.